We're live now. So hi everyone for this Trainee Talks Connectomics session. It's my pleasure to introduce Farhanak Haidari. And Farhanak, please take it away.
Yeah, sure. Hi everyone. Good morning. Just as a brief background about myself, I have a background in electrical engineering and medical imaging, and I'm doing my PhD in biomedical engineering at the University of Toronto in Toronto Western Hospital. I will give a short introduction into epilepsy. That's a chronic neurological disorder, and it's the disease that I will be focusing my research on. Epilepsy is known to be to involve around more than 50 million people worldwide, and unfortunately, in 1/3 of the cases, the medical drugs, the drug treatment does not help patients to become seizure free. So the other treatment option would be surgical resection of the epileptic foci. That would be the seizure initiating area, or a neuromodulation. However, still in 1/3 of these cases, still patients will continue to experience seizures. So the accurate identification of the seizure onset zone is very crucial for getting better results of treatment and helping patients to become epilepsy free or seizure free. Epilepsy is regarded as a network disorder. We know that in the brain, different communities or different nodes communicate through coherence and phase synchronization, and communication pathways are shown that are likely disturbed in seizure initiation, propagation, and termination and modulation of activity. So as we all know, brain, as any other network, can be modeled with a mathematical representation or a graph. And this graph representation of the brain can model the structural connectivity between different brain nodes, the functional connectivity, or the causal relations between different brain nodes.
Farhanak, I'm just going to quickly just jump in. If you click on the hide button, there's a thing on front of your slides there. We'd see a little bit more. Thanks.
Okay. Yeah. So to be able to model the brain as a network, and specifically the epileptic network activity, there are different recordings that are being used in clinical settings that can be used to form this epileptic network graphical representation. They include neuroimaging, T1 weighted DTI and fMRI, and also the electrical signals of the brain recorded through intracranial EEG electrodes. And in my project, I will be focusing on using DTI for representing the structure of the brain, and intracranial EEG for representing the function of different brain areas. The objective of my study is to combine the activity information from IEG with the structure information from DTI MRI, and to determine how the propagation of a seizure is constrained by anatomical connections. Evidence of the relation between anatomy and function has been studied comprehensively. And for example, in some studies, it has been shown that the amplitude and latency of the IEG signals, or the cortical evoked potentials, correlate with the number of DTI pathways, and the seizure onset zone was actually marked by the strength of these IEG signals and in DTI fiber pathways. To go further with our proposal, I will introduce you to a physical concept, which many of you might have heard before about it. We know when a physical object is driven at certain frequency, it vibrates. And these distinct patterns of vibration are called harmonics. You can see at the right-hand side that when, for example, a metal plate is driven at different frequencies, we can visualize these different patterns of harmonics by putting some sand or salt on the metal plate, or, for example, it can be visualized as the sanding waves that occur in nature, for example, on a water surface. Natural phenomena can be described by a combination of a few basis components or harmonics. And our assumption here is that the vibration patterns of the brain actually give rise to activity patterns that can be observed with intracranial EEG. Recently, in a Nature communication publication, it has been shown that there's connectome harmonics, meaning the harmonics driven from the connectomic or the anatomy of the brain derived from DTI, actually can be used as a means of integration of different multimodal data sets. And so it has been shown that it is a very strong theoretical, it gives a very strong theoretical framework for decomposing the connectome into its spatial frequency content. And a weighted linear combination of these spatial frequency components can be used to model any kind of patterns of activity. In our case, it will be the different patterns of seizure propagation. It has been shown that the repertoire of these connectome harmonics actually change with the different brain states. And for example, under LSTUs, there will be a repertoire expansion of these harmonic brain states. So our hypothesis is that the individual's specific connectome harmonics determined from DTI can predict the pattern of macroscale neuronal activity during seizures.
Fahrenheit, just to let you know, we have 1 minute left and we need to bring in the next speaker at 9:30,
yeah.
okay?
Yes, sure. Thank you. In summary, what we will be studying will be extracting individuals' specific connectome harmonics and then reconstructing IEG signals at different time points based on these connectome harmonics to see how the summation of these harmonics change with the different changes in brain state during seizures and after the seizures. So we're hoping, actually, this is the first of its kind in epilepsy literature, and this novel mathematical framework can be used for linking the brain anatomy and its activity to characterize seizure activity, and ultimately it can give an objective tool to identify the seizure onset zones for clinicians. And thank you for listening. Thanks.
Thanks a lot, Fahrenheit. This is a super interesting project. I really am looking forward to catching up with you to chat about this sometime in Toronto. So connectome harmonics is, really exciting new thing in theoretical neuroscience. So this is a really great idea. Looking forward to seeing where you go with this. Unfortunately, we don't have any time for questions for you right now, but hopefully we can connect with people outside of this session. I'm going to kick you off and bring up Peter. So thanks a lot, Fahrenheit.
Thank you.
Peter, you've got the invite, and Fahrenheit, thanks very much. Hi, Peter. Take it away.
Okay. All right. Everything good with the screen?
Yep, looks great.
Good. Okay. Hey everybody, hope you've been enjoying the conference. Firstly, I'd like to thank you for choosing to hear from me. I'll be talking a bit about the ability of whole brain effective connectivity from resting state fMRI discriminate between LSD and placebo conditions. In the past, it's been stigmatized. LSD is now being studied as a potential treatment for major depression disorder. As connectomics researchers, we might wonder whether the efficacy of LSD as an antidepressant treatment could be predicted by looking at a patient's brain connectivity. One route to answering this question would involve first looking at whether the different subjective effects of LSD can be predicted from brain connectivity. Before answering that, we ought to see whether we can correctly classify LSD or placebo using brain connectivity as classification features. We've attempted to answer this preliminary question by analyzing whole brain BOLD data, courteously shared by our colleague Felix Mueller in Basel, from resting state fMRI experiment in which patients were measured twice, one time after taking LSD and another time after taking placebo. Our first question isn't quite specific enough. We need to choose a suitable connectivity measure. To this end, we compared the predictive power of both functional connectivity and effective or directed connectivity. We also wanted to investigate the features which contribute most to discriminating between the conditions, since they may lend some insight into proposed connectivity-related neural mechanisms underlying the effects of LSD. Here again, we compare the connectivity measures. Some important information may be provided by one, but not the other. As I describe our analysis pipeline, you might notice that it resembles generative embedding, if you're familiar with that method. The imaging data provides the input, from which we compute functional connectivity and, enabled by regression dynamic causal modeling, or RDCM, acquire whole brain effective connectivity. The region-to-region correlations and connection strengths will act as features on which we'll train machine learning classification models, which classify the connectivity datasets by condition as LSD or placebo. Using cross-validation, we can gauge the accuracy of these two classification models, one for FC and one for EC, and compare their performance. It's important to note that we acquired effective connection strength parameters over whole brain networks with regression DCM, a newer variant of dynamic causal modeling. DCM assumes that the activities of different brain regions depend on one another in a manner that can be captured by a connection strength parameter matrix, seen here in yellow type. DCM posits that these network dynamics can be inferred from BOLD signal data. Regression dynamic causal modeling, or RDCM, was introduced in 2017. Roughly, RDCM is a modification of DCM by opportune use of approximations and choice of coordinates. RDCM addresses DCM's primary limitation. While RDCM and DCM provide models of comparable quality, RDCM is much more computationally efficient and is capable of modeling networks of over 100 regions. As a result, a more exploratory manner of analysis is possible through RDCM. This is especially useful for resting state data like ours. The results shared today were found using random forest classification models. Random forest conveniently allows us to look at the most important features to the classification. In our case, these are the connections which are most discriminative between the conditions. We found that the classifiers trained on FC and EC yielded comparable performance, which was high at around 89% balanced accuracy. Seems like a promising answer for our first question, but there is a caveat which I'll discuss in a minute. We found that using FC and EC yielded different rankings of feature importance, but that the highlighted connections in both may be interpretable in the context of previous results. The lavender colored bar corresponds to the only connection shared between the two top 10 rankings, which is the bilateral connection between occipital poles. Here we can see the top 200 most discriminative connections. Still not much consistency, However, this is only the top 2 and 4% of the connections considered. When we look at connectograms of the most discriminative connections, we do see some common patterns. We won't seriously interpret these results today, but our guess is that they correspond to altered subjective experiences influenced by LSD. For example, altered connections to occipital areas may reflect visual hallucination effects. Altered connections to frontal areas may reflect altered self-concept, reasoning, or emotional processing. And altered connections to the thalamus may reflect the proposed information gatekeeper role of the thalamus during LSD. Note that we have 2 connectograms for effective connectivity to show connection strengths corresponding to both directions between region pairs. To conclude, we found that LSD and placebo could be classified with about 90% balanced accuracy using brain connectivity. Importantly, LSD causes changes in physiological variables like heart rate, temperature, and blood pressure. Since the measured brain connectivity is likely influenced by these variables, it's likely that our performance is helped by changes in those variables. To answer a question, we would want to look at the extent to which connectivity, independent of these variables, reflects the effects of LSD. We're working on a solution for this. In comparing the choices of FC versus EC, we found that they yielded equivalent classification performance. Clearer differences may be revealed when we correct for the covariates. Though using whole brain effective connectivity did not improve classification performance, that it was comparably effective supports the validity of the approach, which is implemented in the context of LSD research for the first time here. So far, it's hard to tell whether the most discriminative connections can be interpreted in the context of proposed neural mechanisms of LSD. This is another area for which conclusions should be reserved until after we correct for physiological covariates. However, from these results, it is clear that using different connectivity measures yields different information, since the top most discriminative connections are different. We did get the sense from the connectograms that, more generally speaking, the 2 methods appear to be consistent. Perhaps this suggests that at high resolution, directionality in connectivity becomes more informative. This concludes my talk for today, and I'd like to thank everybody that supported this research, and thank you guys for listening.
Thanks a lot, Peter. That's really cool work. I'm going to multitask right now, which is I'm going to invite our next speaker on the screen to get his slides ready while we do a quick question for you, Peter. Does that sound good? So Rejwan, you'll be getting an invite. And Peter, I have a question for you, which I posted in the box just there. And the question was, so you didn't comment too much on the kind of specific patterns, but I just wanted to kind of go in that direction a little bit. So are there like any major trends that you could speak of there in terms of, you know, for example, like increases versus decreases with the psychedelics is the first thing that comes to mind. You know, any kind of structure in those patterns that you kind of, you know, show up strongly?
Yeah. As opposed to simply the feature importance, you mean? Actual increases.
Yeah, well, maybe like the directionality that is picked up by the features. So, and while you're answering that, Rejwan, you can just pull your slides up and get ready to go.
Yep, so we also did a classical steps analysis to investigate directionality. And that's kind of the norm for other literature in this area. So, for example, the interpretation about the thalamus gatekeeper role, we would do the exact same classical stats analysis as that previous paper to investigate directionality. But for now, like I said, the results are relatively preliminary. So when we get more reliable results, we'll be able to make stronger claims and include directionality as a component of that.
Right. Yeah, really great stuff, Peter. We're going to let you go right now, but the regression DCM tool seems like a really powerful technique for the 21st century of neuroimaging. So looking forward to seeing where you go with that. Okay, thank you, Peter. Rejwan, please take it away.
Thanks. So I've just started my PhD. So right now what I'm working on is looking at attractor neural networks and seeing if we play with certain parameters in which we set up the system, how does that affect the retrieval quality when it comes to storing and retrieving memories? And moreover, what does the transition look like when you go to the memory capacity and beyond? So the two things that we want to determine is the capacity and the order of the transition, because there are two cases that occur when you go beyond the memory capacity. Either the retrieval is good until the memory capacity, and then there's a sharp drop to not being able to retrieve any memory, or the retrieval will smoothly drop down to not being able to retrieve any memory. And as like in physics, you can interpret this as a first or second order phase transition, one being sharp, the other one being smooth. So the parameters that we have to play with, and it's not exclusive to these ones, is the pattern distribution. So the distribution that gives you the, should we say, firing rates of the patterns and the probabilities of getting which ones. The mean of these distributions, so what's the mean activity that you get, the number of neurons, of course, the connectivity, which is defined as the number of connections going into a neuron, which is the same for every neuron in the system, and the number of patterns stored so that we can assess the memory capacity. So the distribution, I won't get into too much detail on this actually, but the distributions are constructed so that their first and second order moments are given by this A, which represents the mean activity. And then just looking at the first probability distribution, as this is what I'm on right now, you can see there are two delta functions, one representing no activity and one representing, should we say, firing at maximum activity. And you also have this variable f, which based on the construction is given by A. So the fraction of active units is given by this variable A, the mean activity. So in terms of generating the system, I guess the most important thing is that we have this equation that gives us the synaptic weight matrix, which tells us what the strength between the connections between different neurons are. And this is sort of prescriptive. It says if you have a certain number of patterns, this is how you construct the matrix, the synaptic weight matrix, so that in terms of retrieving memories dynamically, this will give you the optimal performance. In terms of the Hebbian rule, this is contrasted with, because this is a local learning rule, in the sense that any changes you would have in the weights are reflected by the firings of neurons next to each other, as opposed to global learning rules like backpropagation, for example. So the activation function that we use is the threshold linear activation function, which is essentially the ReLU function in machine learning. So this is basically where you have a threshold, and if all the input going into a certain neuron is higher than the threshold, then it will have a positive activity, and it's scaled by this factor G called the gain. And if the summation of all the inputs from the other neurons isn't greater than the threshold, then it will not have any activity. But there is a problem here, because this means that the activities are unconstrained. Technically, based on these equations, you could have an infinite activity. But considering that for now we're looking at binary patterns, that means we need to constrain the activities around 0 and 1. And the way this is done is through a rescaling. We essentially dynamically, should we say, push the network state, the activities of each neuron, so that their distributions are identical to the pattern distributions, or as close as possible. So what this results in is trying to find an optimal value for theta, which is the threshold. And then through that, you can work out the gain through some simple rearrangement of equations. I won't get into too much detail with that, but that's the general idea. So the next thing we need to do is we need to look at what the general shape is of the graph that represents, as we increase the number of patterns, what happens to the retrieval quality. And I just call this an overlap alpha plot. And the idea is you initialize the system so that it's in one of the pattern states, and then you just propagate the dynamics over time and rescale every time step, so changing the threshold and the gain. And you keep repeating this until the overlap is stable for long enough. So stable, I've sort of numerically chosen, and long enough I've numerically chosen. So it's sort of whether the overlap doesn't fluctuate too much and below a certain bound, and long enough so that it has to be within this bound for, say, 20 epochs. And the overlap equation just gives you a certain way of looking at how close the pattern state is to the network state at the current time. So what you can do is you can look at what the overlap values were over time. And just looking at 100 epochs, you can see that if we have 10,000 neurons, which is fully connected, with 19 patterns, which is a very small amount, you can see that the patterns are retrieved almost perfectly. But as you increase the number of patterns, you see that there seems to be a sort of bimodal behavior, in the sense that some patterns remain where they are and they're retrieved almost perfectly, but some aren't retrieved at all, or their retrieval quality is very low, which represents them not falling into the attractor that represents that pattern state, maybe a mix, maybe something completely different. And so when you go beyond this capacity, you can see they all go away from the pattern state, which is represented by a low overlap. And if you work out the mean of each of these plots when they're all stable, which isn't shown here, you have to keep going. You use that as one point of this overlap alpha plot, and then when you continue this,
Rejwan, just to interrupt, sorry. So we've got another
Sure.
1 minute left, and then so if you could wrap up, and then we'll take a quick question if time.
Okay. So the idea is that you generate this plot, you fit the plot with a logistic function, and through that you can work out from the parameters of the fit what the midpoint is, which represents the capacity, and the slope represents how sharply it drops. And then you can play with that to get other things. But I guess I'll just wrap up there then.
So great. Well, so thanks, Rejwan, for the super interesting talk. I'm going to do the same thing of multitask with the changeover and asking a question. So I put my question just quickly in the box there, which is, so why exactly can you not use a saturating activation function here, which would be kind of the standard way of dealing with that explosion issue that you described?
So you mean in the sense that if the activity was over a certain amount, it would just stay constant, essentially?
Yeah, like a sigmoid, right? Which is, in neural and in ML modeling, there's sigmoids everywhere, as we know.
Well, part of the motivation is that this sort of linear activity is related to the activity of pyramidal neurons. That when you inject a current, the response is sort of linear, and it represents a very simple way of getting different behaviors of the system that are easy to work with analytically, and also it's sort of one step into biological plausibility from 1 and 0 step functions.
Yeah, I understand that. Excellent. Well, thanks a lot. This is super interesting work, Rejwan. Sorry to cut you off a little bit early there. So we'll give you the boot. See you around. And Johan, you are last, and we're just barely doing okay for time. So please take it away.
Awesome. Can y'all hear me?
Yeah, hear you great and see the slides great.
Perfect. So thanks for the invitation. My name is Johan Yi. I'm a PhD student under Jason Lurch here at U of T and at the Mouse Imaging Center at SickKids. So I'll be talking about structural covariance, which, if you're not familiar with what that is, it's essentially a correlation or a covariance between the structural properties of a pair of brain regions. So those properties could be things like thickness or structural volumes. And it's a group-wise measure. So to see this, I've plotted the volume of one region against the volume of another region. Each point here is a subject. And for this pair of brain regions, you see that there's a correlation in this population. And then you can do this for every pair of brain regions and represent your resulting structural covariance network using this classic matrix where each row is a region, each column is a region, and the intersection is a color, which just tells you how strongly these two regions correlate or couple together in their volume, or in this case, in cortical thickness. So it turns out that structural covariance is very similar to structural connectivity. So in a study in 2012 by Gong et al., about 35% of all the edges in a structural covariance network had an underlying fiber tract connecting the two regions. In an earlier study by Jason Lurch and others, they found that the cortical thickness of Broca's area correlated strongly with the cortical thickness of Wernicke's area. So these two regions are part of the language network. They're structurally and functionally connected. And so the idea is that maybe population level variation in something like language fluency would explain the correlated variation between both of these regions. So this has led people to think that connectivity is really driving the formation of structural covariance. So you can think of neuronal activation in a brain region can cause processes such as synaptic remodeling to happen in those regions. And from animal studies, we know that these processes can result in volume changes that are detectable through MRI. And so if two regions are connected, these processes can happen in synchrony in a correlated manner, which would manifest itself as a covariance in the structural measures that you see. So to understand this link between structural covariance and structural connectivity, we went to the mouse. So all the data I'm going to talk about was obtained at the Mouse Imaging Center here in Toronto. The setup that we have is a 7 Tesla magnet. We can scan up to 16 brains at a time by putting them all in this basket of RF coils. You put it in the scanner, wait about half a day or do it overnight. At the end of the night or in the morning, you get beautiful 3D whole brain MR images at an isotropic resolution of about 50 micrometers. So once you get these structural images, well, at the end of it, we want volume. So we use a process called deformation-based morphometry. To show you how this works, I'm showing you 4 coronal slices through 4 different mouse brains. And what we do is we align all of these images to a common template or a common brain average. To show you this alignment, I'm going to put on a series of grid lines. And when you warp the brains to the average, you can see this warping through the grid lines shifting. So the idea in deformation-based morphometry is that this warping process encodes volume information. So you can see this if you look at the top here. If a part of the brain has to shrink to meet the average, it means that that part of the brain started off bigger than the average. Similarly, if a part of the brain had to expand to meet the average, that part of the brain was smaller than the average. So in this manner, we get quantitative information on local volumes for every part of the brain for every single subject. And from that, we can compute structural volumes. And then for a population of mice, we can compute the ultimate structural covariance network, which sort of looks like this. I've labeled all the rows and all the columns using colors to denote which region you're looking at. And if you're familiar with mouse neuroanatomy, I've also colored those same regions here on the right. So we compared structural covariance first to structural connectivity. The connectivity data was obtained from the Allen Institute. Essentially, they conducted a series of viral tracing experiments in which they injected a whole bunch of different regions in the mouse brain with a viral tracer. The tracer goes interrograde, it doesn't cross synapses. So it's really a measure of direct neuronal connectivity. And what we see is some overlap, it's pretty weak. It seems that structural covariance is sensitive, but is not very specific to connectivity. More striking, though, was an overlap between structural covariance and networks built from gene expression data. So at the bottom, what you see is a network of transcriptomic similarity. So essentially, the colors here represent how similar two brain regions are in their gene expression profiles. So in some sense, it's a measure of the intrinsic properties of brain regions. And if you think about it from a developmental context, it's you can think of it as a measure of how do two brain regions just intrinsically develop together due to genetic factors. And from this, we see a striking overlap, at least more so than with connectivity. But we really wanted to look more deeply into this notion that connectivity may be driving structural covariance. And so we went to mutant mice. It turns out in the mouse world, we have a mouse strain called a BTBR mouse that doesn't have a corpus callosum. So to us, this was a perfect model to assess what structural covariance looks like in a model of disconnectivity. So our hypothesis is as follows. Suppose you have three regions in the brain, call it a seed region, a target region that's connected to the seed in the same hemisphere, and a target region in the brain in the opposite hemisphere that's connected normally by the corpus callosum. If connectivity drives structural covariance, in mice that have a corpus callosum, if you look at the seed and the ipsilateral target, there's a connection present, and so you expect to see structural covariance as measured through the correlation. So you'd expect to see some level of correlation. In mice that don't have a corpus callosum, if you pick a seed and a target that are connected, so in the same hemisphere, it's connected not by the corpus callosum, you still expect to see a correlation because the connection there, the structural connection is present. If you look at the contralateral side, in between, if you look at a seed region and a target that's connected normally by the corpus callosum, in mice with a corpus callosum, again, connectivity is present, you expect a correlation, but in the mutant mice that don't have a corpus callosum, you expect there to be a weak correlation or no correlation at all. So just to recap, our hypothesis is that when you have connectivity, you expect there to be high correlation between the volumes of these regions, and when you have no connectivity, you expect the covariance to be low. And so we did this actually using not just one set of seed and target regions, but 130 different sets. And when you compute these correlations for all of these different regions, you actually find that this does not fit our hypothesis. If you look at connected regions and unconnected regions, it looks like there's almost no difference at all. In fact, the unconnected regions tend to have almost a slightly higher structural covariance, mean structural covariance value. So essentially that tells us that structural connectivity isn't required for structural covariance. And from the first little bit that I talked about, it seems that it's structural covariance or structural connectivity is less similar to structural covariance than other types of networks, such as gene expression similarity.
So thanks very much, Johan. This is really interesting stuff. I'm going to jump in with a quick question, and then we'll move on to Carmen's session 10. So, well, firstly, the first result you had is surprising, right? That there's like a low correlation with the Allen Institute anatomical connectivity, because those correlations are quite high in human data. But my question is, so basically, would you expect the same thing? Can you do the same experiment for a non-callosal intervention? Because the hemispheres are kind of curious, right? They kind of develop symmetrically, and it's kind of hard to tell whether the things that make one part in the left similar to its homologue in the right is a connection, or is it they're just kind of symmetrically developing?
Yep. So to answer that, I'll answer that in two different ways. The first way is that's the reason why we picked 3 different regions, a seed and 2 targets on each side, as opposed to just a seed and a single target. So that gets rid of the hemisphere effect. And we've also done other modeling to show that it's not just something like transcriptomic similarity that's driving this, which you would expect if something was driving the correlation in both hemispheres at the same time. The other part of the answer is that we've also looked at a different mouse model in which there's no connectivity between other brain regions, and we sort of see the same thing, that structural covariance doesn't change.
Right. Yeah, super interesting stuff. All right. Thank you, Johan, and thank you everyone for your talks today. So just round of applause from me and everyone else attending, I'm sure. And we're going to go into the next session now. So I hope everyone enjoys the rest of the conference. Bye. Thanks a lot.